Hello, I'm Professor Sims, and in this video I will discuss the heart. This is the third in a series of 10 lessons held as part of my Anatomy and Physiology 2 online laboratory course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. In this unit, we'll describe the size, shape, and location of the human heart. We'll define and locate the pericardial layers, identify and describe the interior and exterior features of the heart using models, images, and dissection specimen. We will describe the path of blood flow through the heart, differentiate the unique features of cardiac muscle cells compared to skeletal and smooth muscles. So first and foremost, the heart, it acts as a muscular pump that circulates blood throughout the body. And this ensures that oxygen and nutrients are delivered to tissues while carbon dioxide and waste products are carried away for removal. The heart is also important for maintaining blood pressure, keeping it strong enough to move the blood efficiently through the vessels. And it uses valves to control the direction of blood flow, ensuring that there's a one-way flow and no backflow. Finally, the heart supports two major circuits. The first one's called the pulmonary circuit. Pulmonary circulation moves blood to the lungs for gas exchange, and then systemic circulation delivers oxygen-rich blood to the rest of the body. The heart has 4 chambers, 2 atria on top that receive blood, and 2 ventricles on the bottom that pump blood out of the heart to the lungs and the body. Valves are located between the chambers and at the exits of the heart. Valves act like one-way doors that prevent backflow and keep blood moving in the right direction. The major vessels connected to the heart include the vena cava, which brings oxygen-poor blood into the right atrium, the pulmonary veins, which return oxygen-rich blood from the lungs, the pulmonary trunk, which carries blood to the lungs, and the aorta, which sends oxygenated blood to the body. The heart wall has 3 layers. First is the endocardium. This is the smooth inner lining. Then you have myocardium. That's the thick muscular middle layer. And the epicardium, which is the outermost layer. This also forms part of the pericardium. The heart is made of cardiac muscle, which is striated and branched, and it has cells connected by intercalated discs that allow for fast coordinated contractions. The heart beats in rhythm thanks to a built-in conduction system that includes the SA node, AV node, bundle branches, and Purkinje fibers. And then these structures all work together to coordinate each heartbeat and ensure efficient pumping. The right atrium and left atrium are the superior chambers, which means that they sit at the top of the heart, and these chambers are responsible for receiving blood that is returning to the heart. The right atrium receives deoxygenated blood from the body via the vena cava, and the left atrium receives oxygenated blood from the lungs through the pulmonary veins. And these are separated from each other by the atrial septum. This is a thin wall that keeps oxygen-rich and oxygen-poor blood separated so they won't mix with each other. The right ventricle and left ventricle are the inferior chambers, which means they're on the bottom and below the atria. These chambers pump blood out of the heart. The right ventricle pumps deoxygenated blood to the lungs through the pulmonary trunk, and the left ventricle, with its thicker wall, pumps oxygenated blood to the entire body through the aorta. And these chambers are divided by the interventricular septum, which is a thick muscular wall that separates the 2 ventricles. There are 4 major valves that ensure one-way flow of blood through the chambers. First, there is the atrioventricular valve, or the AV. The tricuspid valve is located between the right atrium and left ventricle. The mitral valve, which is also known as the bicuspid valve, is between the left atrium and left ventricle. Both AV valves are anchored in place by the chordae tendineae, which are strong tendon-like cords that attach the valve leaflets to papillary muscles on the inner walls of the ventricles. When the ventricles contract, the papillary muscles also contract, pulling on the chordae tendineae, and this prevents the valves from everting or flipping backwards into the atria, which would allow backflow. And then we have the semilunar valves, which control blood flow going out of the ventricles. The pulmonary valve is located between the right ventricle and the pulmonary trunk, which leads to the lungs. And the aortic valve is between the left ventricle and the aorta, which carries oxygen-rich blood to the body. And these valves open and close based on pressure changes, and they ensure that blood moves through the heart and into the circulation without flowing backwards. I keep reiterating the flow of blood because it is important. We're going to talk about vessels of the heart, and when we do that, we're going to be referring to major blood pathways that carry blood to and from the heart, where it's going to be important to remember that arteries are always carrying blood away from the heart, regardless of whether it's oxygen rich or oxygen poor. And the pulmonary arteries are an exception to the typical oxygen rule because they carry deoxygenated blood from the right ventricle to the lungs for gas exchange. But typically, blood leaves the heart through the pulmonary trunk, which quickly splits into the right and left pulmonary arteries, and this is directing blood to each of the lungs. And then on the other side, the aorta is the largest artery in the body, and the aorta carries oxygenated blood from the left ventricle to the rest of the body. And it has 3 parts. You have the ascending aorta, which comes directly from the heart, the aortic arch, which gives off branches to the head and arms, and the descending aorta, which branches off to supply blood to the lower part of the body. And these arteries are all important for systemic and pulmonary circulation. They help maintain proper oxygen delivery and waste removal in the bloodstream. The other thing we need to keep in mind are the veins. Veins are responsible for bringing blood back to the heart. You have the superior vena cava, returns deoxygenated blood from the upper body, including the head, neck, and arms, into the right atrium. The inferior vena cava brings the deoxygenated blood from the lower body, like the legs and abdomen, also into the right atrium. Coronary sinus is a little bit different. It's the vein that collects deoxygenated blood from the heart muscle itself, or the myocardium. It drains it into the right atrium. And then finally, the pulmonary veins are also a bit special because they carry oxygenated blood from the lungs back to the left atrium. And there are right and left pulmonary veins on each side. And then together, all of the veins complete the circulatory loop by returning blood to the heart. And then from there, they are going to be ready to be sent back to the lungs or the body again. We discussed briefly the layers of the wall. Let's talk about those in just a bit more detail because it's important for you to know each layer of the heart wall and what their unique structure and functions are going to be. So the endocardium, again, that's the thin, smooth inner lining that coats the heart chambers. The endocardium is composed of simple squamous epithelium and connective tissue, and this makes a slick surface that reduces friction as blood is flowing through the heart itself. Importantly, the endocardium is continuous with the endothelium of the blood vessels, and this provides a seamless lining throughout the circulatory system. The myocardium it's thicker. It's the thick muscular middle layer of the heart wall, and this is made of specialized cardiac muscle tissue that contracts in order to pump blood. So the myocardium is the big muscle part that contracts the heart. Myocardium is thickest in the left ventricle because this is the chamber that has to generate a lot of force to send blood through the entire systemic circulation. Then you have the epicardium. This is also known as the visceral layer of the serous pericardium. So the epicardium, it's made of connective tissues and epithelium, and it contains important structures like the blood vessels, nerves, and fat, and that helps to nourish and protect the heart muscle. Surrounding the heart is the pericardium. Pericardium is a double-walled sac that provides both protection and lubrication to the heart. The fibrous pericardium is the tough outer layer that anchors the heart to the diaphragm and the surrounding structures, and this keeps the heart in place within the chest. Inside of that is the serous pericardium that and it has 2 layers. The parietal layer lining the fibrous pericardium and the visceral layer, which is the epicardium itself. And then between these 2 layers is the pericardial cavity, which is filled with fluid that reduces friction as the heart beats and moves. So let's have a look at that in this figure here. From the inside out, you see the endocardium, the myocardium, epicardium, and you can see there's very big differences in not only their thickness, but their density and what types of cells they're composed of. This is a more detailed look at the pericardium and the pericardial sac and all of those layers that we just talked about. You see you have the sac on the outside, and then you have this layer just inside where all of the blood vessels are. Let's look a bit more at cardiac muscle and the specialized tissues that make up the heart wall. Cardiac muscle cells are striated and branched, and they give the tissue both strength and flexibility. And this is the type of muscle that is only found in the heart. The cells are connected end to end by structures called intercalated discs. Intercalated discs contain 2 key components, the desmosomes and gap junctions. Desmosomes are kind of like little anchors. They hold the cells tightly together during the contractions of the heart. And then gap junctions create little channels that allow ions to pass directly between the cells. And this allows the heart muscle to contract in a coordinated and synchronized way, which is essential for a good heartbeat. Each cardiac muscle is typically just has one centrally located nucleus, and because the heart is working continuously without any rest, these cells have numerous mitochondria that provide the high energy that's needed for this constant contraction and movement. And understanding all of these features, it helps to make it clearer why cardiac muscle it's so specialized and it's so well suited to the role of pumping blood. Here's a figure that is showing all of these features in more detail. Notice how there are numerous mitochondria in all of these cells. And this is showing the intercalated discs that are located between these striated and branched muscle fibers. This here is a gap junction. You've seen gap junctions when you studied like cell signaling. This allows for ions to pass from one area to the next. So it's essentially how the heart muscles can all talk to each other and synchronize. The heart's pumping action, again, it's carefully synchronized and it's controlled by its conduction system. This is a network of specialized cardiac muscle cells that coordinate with each other. So let's dig into that a little bit more. You have the sinoatrial node, the SA node. This is in the right atrium, and this is the node that is known as the natural pacemaker because it initiates each heartbeat, and it does this by generating an electrical impulse. And then the impulse quickly spreads through the atria and it causes those to contract and then reaches the atrioventricular node, the AV node. This briefly delays the signal just long enough to allow the ventricles time to fill up with blood. And then from the AV node, the electrical signal travels down into the bundle of HIS, which splits into the right and left bundle branches that run along the interventricular septum. And then finally the electrical impulse is distributed by the Purkinje fibers throughout the ventricular walls, and this triggers a coordinated contraction of both ventricles, and this pumps the blood to the lungs and to the rest of the body. Let's walk through these electrical events using this figure here. So again, the cycle begins with the heartbeat at rest until the SA node fires, and it generates an action potential that spreads across the atria, causing the atria to contract. Then the impulse will reach the AV node where there's a brief 100 millisecond delay. And this pause is important because it gives the atria time to empty all of the blood out into the ventricles before they contract. The signal then travels down into the AV bundle, the bundle of HIS, through the right and then left bundle branches and into the Purkinje fibers. And it also reaches the right papillary muscle early, which helps helps to coordinate valve closure and pressure as the pressure is building. And then the signal spreads through the ventricular muscle fibers, triggering ventricular contraction and completing the heartbeat. And all of this activity can be seen in an electrocardiogram or an ECG. You also may have heard it called an EKG, but this is used to track the electrical signals, and it gives us a visible tracing of the heart's rhythm and timing tied to each mechanical event. So, as usual, make sure you do the pre-lab stuff. Read chapter 19 in the textbook. This is covering the structure, function, and physiology of the heart, a good foundation. Then you'll look at the intro and background sections of chapter 3 in the lab manual. Complete the pre-lab exercises, and then use those answers to do the pre-lab quiz on Moodle. And then for lab exercise 3.1, your task is to identify and label the right atrium, right ventricle, tricuspid valve, the two chordae tendineae attachments, the left atrium, left ventricle, bicuspid, also known as mitral valve, and papillary muscle, and the superior vena cava, the three major arteries branching off the aortic arch, the brachiocephalic artery, left common carotid artery, and the left subclavian artery. This diagram is showing the anterior or the front view of the heart, and just like you'll see on the model when you're looking at it from the front, from this view you should be able to identify the right atrium and right ventricle on the left side of the diagram, the left atrium and left ventricle, which is actually on the right side of this diagram. The superior vena cava is entering the right atrium from above, right here. The aortic arch, giving rise to the three major arteries, brachiocephalic artery, left common carotid artery, and in the left subclavian artery. Here it is right here. Left subclavian, left common carotid, brachiocephalic artery. Also visible from the anterior view are portions of the pulmonary trunk, coronary sulcus, and possibly the auricles of the atria. So use this as a reference for identifying the external features and blood vessel connections on the front of the heart. Then you have the posterior view or the back. From this angle, you should look for the left atrium, which is much more prominent on the posterior side. Then openings of the right and left pulmonary veins entering the left atrium. The inferior vena cava entering the right atrium from below. See it right here. The coronary sinus, which is a large vein that drains blood from the heart muscle into the right atrium, and the posterior walls of the left and right ventricles. This view is good for recognizing how blood returns to the heart, especially oxygenated blood coming from the lungs via the pulmonary veins. For lab exercise 3.2, you'll be looking again at histology slides that show various structures within the heart wall. You'll use the figures 3.4 through 3.7 in chapter 3 of the lab manual, and you'll want to be able to label and identify cardiac myocytes, intercalated discs, myocardium, endocardium, endothelium, and Purkinje fibers. These are all going to be visible under the microscope, but they can be difficult to recognize at first. So let's walk through two labeled diagrams that will help you match what you see on the slide with the correct terms. Cardiac muscle is striated, branched, and has single central nucleus. Intercalated discs connect these cells and help synchronize contraction. So the intercalated discs are going to be connecting the striated muscles together. Then you have Purkinje fibers. They're going to appear paler and often have two nuclei because they conduct signals. They don't contract. They are sending electrical signals. Endothelium is the smooth inner lining. It's part of the endocardium, as we discussed, and it's going to appear as a much thinner layer of squamous cells, and you want to use those kinds of cues to identify these structures on the histology slides. This is a sample histology slide, and you can see how it can be kind of difficult to see these layers unless you know what you're looking for. This here is the endothelium, and it's a thin, flat layer of squamous cells, squamous epithelial cells, and beneath the endothelium is the endocardium. The endocardium includes endothelium and a layer of connective tissue that gives more structural support. And then the myocardium is beneath the endothelium, beneath the endocardium. And this is the much thicker muscular layer that makes up the heart. Let's zoom in a little bit more. We'll look at cardiac muscle tissue and Purkinje fibers in a bit more detail. Your Purkinje fibers are here. They are going to appear larger and paler than the surrounding muscle fibers because they contain fewer proteins. And they often have 2 nuclei, and they are conducting electrical impulses, not generating force. So they're not going to be like these big, thick cardiac muscles. They're a lot smaller, they're paler, they have less protein. They're used for conducting electricity. They're also embedded in loose connective tissue just beneath the endocardium. And then you have over here this slide, or this image of a slide, is showing branching cardiac myocytes. It shows the striations and the central nuclei. You have intercalated discs, which again are these like kind of darker stained parts here. Intercalated discs, they kind of look like lines in this slide, whereas in the last slide they looked a lot more like these nuclei, so don't let that fool you. But this is where one cardiac muscle cell is connecting to the next, and these are the structures that allow for mechanical and electrical connections between these fibers. And it helps they look like little insignificant lines, but these are the things that allow for the coordinated heartbeat. So they're actually really, really important. For exercise 3.3, you will be looking at the path of blood flow through the heart using an interactive online simulation from Nova. So you'll want to go to the website, and then as you click through, there's 6 steps of blood flow. And through each of these steps, you want to pay close attention to the direction of flow, which chamber, which valve, which vessel the blood is entering and exiting, whether the blood is oxygenated or deoxygenated at each step. So you will actually track this by observing the color changes from blue. Blue means low oxygen to red. Red is high oxygen. This activity helps to reinforce the relationship between the heart structures and functions. For example, why the left ventricle has the thickest wall, and how the valves, how do they maintain one-way flow? And then once you have finished all 6 of those steps, be sure to answer the related questions that are in your lab manual. That's going to help you to solidify your understanding of the full circulatory loop, including both systemic and pulmonary circulation. Now for the next section, if you are attending lab in person, now I've designed this for an online course, but some people might be using these slides for an in-person lab. If you were doing it in lab, you would have a preserved sheep heart, dissection tools, reference diagrams, and you would begin with the external observation, noting size, orientation, and visible vessels, including aorta, pulmonary trunk, and coronary arteries and veins. Then you make your incision, longitudinal incision to open the chambers, identify the right atrium, right ventricle, tricuspid valve, chordae tendineae, left atrium, left ventricle, bicuspid valve, interventricular septum, aortic and pulmonary semilunar valves. And then, of course, if you're an online student or if you are a face-to-face student that doesn't have specimen available, you don't have the hearts, you don't have to worry, because you can still complete the activity by just looking at the labeled figures to follow. So let's have a look here. This is the anterior view of a preserved sheep heart. For example, you can see the pulmonary trunk here, the right ventricle, left ventricle here again, pulmonary trunk. So you can see all of the external structures that we discussed in this figure. This is the same sheep heart over here. It's just the posterior view. So this should look pretty familiar since you have already identified all of these on the models, you've got your aorta, pulmonary veins, right ventricle, vena cava, inferior vena cava, right? This here is just a heart that has, I think these are pencils or rods that are filling in for veins and arteries to help kind of show you how it would look if it had blood pressure in the heart. These are some views from the interior, so this would be after you make your incisions. See the right ventricle here, pulmonary, papillary muscle. It's very small and hard to see, but these are the tendini, tricuspid valve, superior vena cava. Again, these preserved specimens sometimes it's really difficult to see things. Sometimes you get really lucky and it's just all kind of laid out for you. Like here, it's easier to see some of the blood vessels in this one, for example. The external blood vessels are going to be easier to identify sometimes than it just kind of depends. Also, smaller hearts, it's more difficult to find all of the structures on the smaller heart than it is, say, if we had like a cow heart. This here is kind of like it's a black and white image, but the contrast has been cranked way up so that you can see some of these little smaller bits more easily. And this kind of stuff too, it's like difficult to tell the interior blood vessels from each other unless you really have nailed down like the pathway, because you can use the ventricles and larger structures as your reference point. Like if you know that this artery goes from this ventricle or from this aorta to this ventricle, right, you can kind of trace it that way. Okay, then, well, that's going to be it for Lab 3. Thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics, and leave your questions for me in the comments below.